called a palliative treatment, which is to make the patient more comfortable. And we thought, well, if it affects therapy, that would be important to know. So for our model system, we adapted something that had been used to study lung cancer before. So this is a 6-well plate. It's fitted with stoppers that fit into the top of each plate. And then we have a syringe and a pressure gauge connected to the top. And we're basically just providing an overhead pressure, so pressure head on top of the cell culture media and cells. This is what the actual device looks like with the different, actually, I guess it's 12-volt plate, sorry, with the different stoppers. And this allows us to provide a hydrostatic pressure, which is not necessarily exactly the same as interstitial fluid pressure, but again, this is the model. So we looked at 3 different cell lines, U87, U251, LM229. These are all established glioblastoma cell lines. But what's interesting is they have different properties. So U87 forms more nodular cancers, LM229 forms more diffuse and spread cancers, and U251 is somewhere in the middle. So that kind of gives us some different phenotypes that we can across. And so this is what we found in our first experiment. So we have 4 pressures that we're testing. 0 is just a dish with no pressure applied. 9 is the physiological pressure that you would experience in a healthy patient. 22 was what we used for a low tumor, so a tumor with low interstitial fluid pressure, perhaps receiving treatment, and a high tumor of 47. And we found this parabolic response in all of the cell lines. What we're looking at is viability. So at atmospheric pressure, our cells are fine, everything's fine. As we begin to apply pressure, we see a lot of cell death. But interestingly, at very high pressure, we don't see cell death, and that suggests some sort of avoidance mechanism. We compared this also to normal cells. So these are astrocytes, there's cell type from which glioblastoma derives, normal human astrocytes. We do see a parabolic response, but there's no statistical significance. So these cells do not respond in the same way as the cancer cells. So this suggests that this is a cancer-specific response. We then asked whether this is apoptosis or necrosis. So necrosis is cell death where like I stab you. And with high pressure, that is a possibility. I could just simply rupture the cell. Apoptosis is controlled cell death. And we found that the majority of cell death that was occurring was apoptosis, not necrosis, which is shown in red. So that suggests that when the high pressure cells avoid death, they're avoiding these apoptotic mechanisms. And this data told us that high interstitial fluid pressure increases the resistance to apoptosis. So that treatment that patients are getting to reduce pressure is probably a good thing. Okay, so we were wondering how this relates to patient data. So with our collaboration with Dr. Otero, we were able to basically pull data from the Allen Brain Atlas, which has data on both normal patients and GB patients, and sort of compare what was happening. The first thing we did is we had to identify mechanosensory genes to figure out which genes might be differentially expressed in patients that had high IFP versus low IFP. And so for our model, we looked at comparing the tumor periphery, where we would expect the pressure to be low, to cells in what's called the perinecrotic region, so very near the center of the core of the tumor. And we compared those 2 sites. And then we had the normal patient data, which we used to identify mechanosensory genes. And then we applied that gene set to the patient data to see which ones were different. So this is just a volcano plot, and the way that this works is anything above a log of 2 difference is considered statistically significant, but it also has to have a p-value of greater than 0.05. So this blue line, so anything in red is a significant gene, and all the gray is not significantly changed. I don't necessarily want to rattle on this too long, but these were the top 10 genes that we identified, and there's a few categories. So green are genes that have to do with neuron signaling. Not surprisingly that those might be pressure sensitive. Yellow has to do with immune response and migration. Blue has to do with cell metabolism, and then at the bottom, we have cell differentiation. So these are some of the pathways that were implicated. But we also went and did a full pathway analysis, where we looked at the upregulated genes and whether they belonged to the family of cell division, apoptosis, and then the mechano at the top are the genes that were different. So we found that many more genes that were different intersected with apoptosis, 81, versus the 9 in cell division, actually 81 plus 5, so 86 versus the 9 in cell division. So this tells us that our finding of differences in apoptosis is supported by data from human patient data sets. And if we look at the pathways that were particularly implicated, we pulled out ones that had to do with apoptosis or crossed with that, and you can see quite a few that were implicated in our study. So we wanted to understand what was causing this change, and our hypothesis was that it was these 3 parts of the pathway. So this, again, is that KEGG diagram, and here's the cell membrane. This is integrin A or integrin B, and our hypothesis was that this was initiated by integrin beta 1. And we said this because we know that any sort of mechano response probably initiates from the cell membrane. There's been a lot of work on mechanotransduction in cancer and other contexts, and beta 1 is a known mediator of that. Beta 1 is upregulated in glioblastoma. That would explain why we might see a different response in cancer versus normal cells. So that's kind of how we formed our hypothesis. And then we proposed that it signaled to focal adhesion kinase and then to AKT. AKT is a mediator of apoptosis. So we thought that sounds like a good pathway. So what we did is we repeated these experiments and we did Western blots for the 3 markers. And for these 2, we also did their phosphorylated amounts. And so this is kind of what we found. And as a reminder, what we're looking for is something that looks like this. We want that parabolic response. It could be inverse parabola because it might be downregulated versus upregulated, but that's what we're looking for. And so with beta 1, we do see that parabolic response. And so we're like, this is good evidence to suggest that that is a molecule that's mediating this. But for FAK and phospho-FAK, we're not really seeing it in the same way that we do with beta 1. And also with AKT, probably not, and PAKT, probably not. So we think this is probably correct, and this is probably not. And the way to really get at that is we're now beginning inhibitory studies where we can specifically inhibit this molecule, or this molecule to determine if the behavior stays or leaves. And so that's something we're in the process of right now. So just kind of want to remind you, this work is addressing this resistance to therapy and trying to understand how cells navigate treatments, how they change their response to overcome therapies that are provided, and whether or not IFP can kind of connect to a therapeutic response. So I now want to talk a little bit about some of the work we've done in diagnostics, which is kind of the other piece of this puzzle. So this is work that I've done over 20 years. There's a lot of people that have participated in this, and we'll kind of march through them as we go. So the idea was when I started my faculty career, I was working on farm dogs, I was a bio person, and I wanted to use them in clinical diagnostics. So initially, I thought maybe it would be good in solid tumor pathology. Turns out not so much because most solid tumor pathology just uses colored dyes, not fluorescent. And I'll talk about that more in the next section. But I was encouraged to apply them to liquid pathology. So this is used in diagnosis of hematological cancers like leukemia and lymphoma. And the primary way that those are diagnosed is via flow cytometry. So the way that flow cytometry works is you have these cells, and I can use flow just on the cell itself without any label and look at its size and complexity, but most people use a fluorescence label in combination with that. So I apply a fluorescence label to the cell, and it moves through a microfluidic channel cell by cell, and there's a laser, and when the laser hits the cell, if the cell has the fluorescence, it will detect it, and if it doesn't, then it will not detect it. And so you get a map, cell by cell, of how fluorescent they are. And you can do this with combinations of 8 or 9 fluorophores. So you can look at lots of biomarkers at the same time. So this is what a typical plot might look like. So here we have blue fluorescence and green fluorescence. So we have two markers that are being used. And any cell that's high on the x-axis has lots of blue, high on the green, has lots of, high on the y has lots of green, and of course, in the different quadrants are plus minus those. So one thing that people will do is they do something called gating. So that's the red lines here, to divide these into positive, negative cells that we can quantify by phenotype. And I can generate a histogram like this and say these are negative cells and these are positive cells. So this is fluorescence intensity and this is number. And I want to point out this is a log plot. So this is 2 logs different from the control, basically in terms of intensity. So that's kind of how you might like read or interpret a flow cytometry plot. So I mentioned leukemia and lymphoma detection, and I put this up just for you to think about. So this is a detection scheme for acute myeloid leukemia, and the blue are the markers. So what I want you to just notice is the number. So in the very first triage that you would do, you have 4 markers on one side, 2 on the other, so 6 markers that I'm imaging for. So this is a scenario where for this disease diagnosis, I need lots and lots of labels, lots and lots of fluorescence colors. And that's something that Quantibots actually do really well because they have a narrow emission bandwidth. So they can easily be discerned, like in orange from the yellow, whereas the fluorophore dye, which have a broader spectrum, do not. So as an example, they have these things called spectrum builders that you can use to build your flow cytometry panel. And you basically type in what are the different colors you plan to use, and then it will plot them on the image. And what I want you to notice is that a lot of these colors overlap. Like these two cannot be used together. I would not be able to tell them apart. So this is really a problem in flow cytometry imaging and in the current diagnostic technology, which uses chemical fluorophores. The spectrum, the amount of width of these bands is very wide, and it makes it difficult to have large panels. One of the things that people do to avoid this is something called tandem dyes, which basically use FRET or forced resonance energy transfer. So it'll have an absorption at one color, and then that dye transfers its energy to another dye and emits at a different color. Those dyes are only stable for about an hour. So a clinician has to make them and immediately use them. And this is a huge pain point in the clinical market. So my question was, could we use quantum dots to address this? The quantum dots are semiconductor nanocrystals. They're about 2 to 10 nanometers in diameter. And in 1999, when I started my PhD, everyone said, they're going to revolutionize biomedical imaging. We're all going to be using quantum dots. But we're not. Why not? Let's get to that. So they have tunable fluorescence based on size. So if I change the size slightly, the color changes. So that gives me a lot of control over the different colors. Whereas for a fluorophore, I have to chemically modify the fluor to get a different color. So it's a lot more difficult to get a color panel. They can all be excited by the same laser. So for the fluors, for each color, I might have a different laser. So I have 9 different lasers to get the 9 different colors. That's time consuming because it takes time for each laser to flash across that cell. Here you could do everything with one. So that will cut time. They also have a narrow bandwidth, which I mentioned, which reduces what's called that spectral overlap, which is where the spectra overlay each other. But I've been working on this for more than 20 years, so this was not an easy journey. So what happened? Why did they not work? Well, one of the biggest issues is solubility. So quantum dots are manufactured in organic solvent. And in order to use them in biology, you have to coat them or somehow transfer them into water. And a lot of the processes for that results in loss of colloidal stability. And what would happen is kind of the processes that people were using most commonly worked well enough. But when I tried to bioconjugate anything to this quantum dot, it would crash out of solution. So those are the dots. So this is showing stability as a function of different buffers. And we've got pure water, which is obviously not biocompatible because it doesn't have stable limit. borate buffer, which is what's recommended by the manufacturer. And then this MES buffer, which is a standard buffer used for EDC bioconjugation, which is sort of the gold standard workhorse. And when I do MES, like they all crash out. That's what the stars are. So all my particles down at the bottom of the dish, not soluble. Well, that's not good work. Another problem is the fluorescence depends on the coating method. So this is from some of our work in nanomanufacturing. We tested 3 different methods for making particles. The particles are made of all exactly the same thing. So we're coating a particle with the same polymer, same materials, and yet when we do it with sonication, it immediately has low fluorescence. When we do it with electrospray, it starts out okay and then goes down. But when we do it with this liquid electrospray method that we developed, it is maintained longer. So a chemist thinks I put these things together and they're the same every time, and an engineer knows that it actually matters how you put them together and what process is used. And so this was a problem as well, because it matters how you make it. Another issue was the charge. So if I'm going to put a nanoparticle in a polymer or coating and try to use this for bio application, I need that polymer or coating to have something I can conjugate to on the outside in order to target the cell. I need to put it, I need to conjugate it to an antibody or a peptide. And in order to do that, I probably need a charged group, like a carboxyl or an amine. And what we found is when you coat them with charged molecules, like amines and carboxyls, the fluorescence is much lower than when you used a molecule that didn't have a charge. Problem 2. And then we determined that that was because the formation is slower when you make them with charged polymers because of charge repulsion. The polymers don't want to come together as quickly. And that allows the organic particle to be exposed to the aqueous phase longer, which resulted in its oxidation. And when it oxidized, it lost fluorescence. And that was actually a mechanism of what was happening here as well. So it took me a really long time to figure all these things out. But we did. And this is partly why it was so difficult to commercialize this technology and kind of bring this into a clinical market. Another is easy to see. So one of the solutions that we came up with was to encapsulate particles and black copolymer micelles. And the reason is a black copolymer is pretty long, and that provides a really thick coating, which should protect the particle. But our innovation was that we were able to encapsulate multiple particles within the same micelle. So even though the particle was it ended up being about the same size as the ones that are commercially sold with really thick polymer coatings, but it's brighter, so there's more particles in it. So here you can see the particles that we made have an intensity of about 40,000. This is over time, just showing you the fluctuations. When we took those same particles and just did a method called ligand exchange, where we took off the organic ligand on the surface and replaced it with a water-soluble one, this is only 4,000, and we have multiple incidents of what's called quantum blinking, which is where they just lose fluorescence for a minute. They look like stars. They fluctuate. It's kind of fun to watch, like record or something. These are the commercial ones that are sold. They're about 9,000, and these are the original organic at about 6. So putting a lot of particles together really ups the fluorescence. Why is that important? It makes it easier to detect low-expressed biomarkers. So I can detect your disease sooner or better stratify a patient population. So it also reduced oxidation because I have this thick black copolymer coating. It's harder for oxygen to diffuse in and damage the molecule. So these are particles that we've exposed to laser of really high energy, like we're basically trying to burn them. And the micelle encapsulated particles on the top, we have 2 different colors, green and sort of like orange. They stay stable a lot longer than the commercial particles on the bottom. And then I mentioned that we have been loading lots of particles inside one core, and we actually can make ones with just one particle and kind of comparing them. Of course, their fluorescence is lower just to show you when, but we also found that as we age these particles, the gray versus the black, they lose about half their fluorescence. And we believe that's because when you have a lot of quantum dots in there, a couple of them can act as like a sacrificial layer, but the ones in the middle are still protected. So as the oxidation is occurring, you're just losing the outside, but you're not losing everything. Whereas if you only have one particle and it oxidizes, then you have them. So we think this is a path forward for commercialization. And we started working with Jeff Chalmers and Miriam Westberg and breast cancer to look at whether or not we could use these to quantify circulating tumor cells. Because at this time I was doing this work, that was all the rage. And we started by labeling leukocyte, which is a white blood cell. And if you ever need to test a cell label, I suggest you start with a leukocyte. Leukocytes express CD45 receptor, and they express it over a huge portion of their cell membrane. So if you can't label CD45 on a leukocyte, you can't label anything. So we thought we'll start with that. And then we labeled BT474, which is the breast cancer cell line. And then we labeled circulating tumor cells that we obtained from metastatic breast cancer patients. And so we were looking for the HER2 biomarker, which is an indicator for patient treatment with Herceptin. It's one of the 3 triage biomarkers for breast cancer patients. We had to develop some image analysis technology to basically quantify the expression of this receptor with our labels. And then we generate histograms very similar to what you would get in a flow cytometer that show the number of particles versus the percent of cells that have that level of fluorescence. So this sort of correlates to the number of receptors. And so what you can see is for CD45, we were able to get a nice histogram. Then we did the HER2 and then the HER2 on the cancer patient cells. For the CD45, we were able to compare it in the literature. So we found about 50,000, and the reported value from flow varies from 25,000 to 200,000, and that's by cell. So this histogram reflects this data well. So we believe another advantage of this particle is it's so small that it's maybe possible to label individual receptors and get a receptor quantification rather than just a plus or minus on the number of receptors expressed. And in doing this, we were able to see some interesting trends in the patient data. So they were looking for circulating tumor cells, and even with initial enrichment, which is what Jeff Chalmers did, 94% of the cells were still not circulating tumor cells. There are other blood cells that were captured. But we did find that the CTC is stratified into 3 groups. So we had a low, medium, and high expressor. And what's interesting about that is it can give the clinician the power to ask whether or not these HER2 therapies are working. Meaning, do they only kill the high expressors, or do they get all 3? And if they only kill one population, maybe they could add another treatment to address the low expressing cells. And we can pull these cells out and figure out what other markers they express. Maybe there's other therapies they could respond to. So this kind of leads into a personalized therapy approach where we look at each patient and how they're responding and then adapt their therapy based on that data. So we started a company to sell these things in 2012. So Christy Melnick is our CEO. Suri Fian was a student in my group who transitioned to the company when he graduated. And then Ms. Ray Uni is the current chief technology officer of the company. So starting a company is really, really hard. Let's start with that. I don't even want to talk about all the issues that we have, but we have them all, I promise you, including our initial CEO embezzled money from us. So we really had every problem, but we have persevered. We have received state, federal, and private funding. We're currently funded by a venture capitalist who is basically all in until we're cash flow positive. We started research use only field in January of 2020. So you can imagine how that went. The customers that we were trying to reach were the same labs doing all the COVID tests. So we weren't even able to get to our customer base. So we immediately pivoted to a second product development, and we have a magnetic version So you can use this for cell separation and quantification at the same time. And our goal has always been analyte-specific reagent sales. What does that mean? So most of the time when you sell something into the FDA, you're either a device or a drug. And then you can sort of be a new drug or a predicate use something. And then if it's predicate use, there's other things like it. So if I'm lead and I'm like Tylenol, it's easier to get approved than if I'm some brand new thing like the COVID vaccine that's never been out there. There's a third category called analyte-specific reagents, though. This is the easiest possible path to FDA approval. And what it is the reagent used in the test. And all you have to do is show that it works the same or better than the reagent currently used. You register with the FDA, you have to have ISO certification, but there's no like approval per se in the classical sense. The downside is every lab that uses your product has to validate it at their site individually. That's kind of how the ASR system works. But that is what we chose to go after because that made the most sense for our product. So this is the data that really shows what we do. So the the grayed out background is phycoerythrin dye control. So this is what they're using now. And I have negative cells and I have positive cells. And I want you to look at the overlap. So the negative and the positive are kind of overlapping. That's problematic because it makes it difficult for me to determine what that patient's status is. There's this population of cells that could be normal underneath the positive peak. So on the right, we have the quantum dots that are titrated at different doses. And you can see that the positive peak is moving out the dose to the right. The first thing I want you to take away is a reminder that this is a log plot. So these are exponentially brighter than what's currently being used. So that makes it easier to detect when you have a very low amount of a transition. But the other thing that's important is the negative peak is not really moving over to the right. So a lot of times when I increase product dose like that, I have non-specific binding, the negative peak is moving too. We don't see that. So that means I can now more clearly discern a negative cell from a positive cell. And that helps me to identify whether a patient is having a relapse or not. And so these have direct benefits to patient therapy. But this took me 10 years. This one graph is 10 years. All those problems I told you about earlier, we have to solve all of them before we make a product that works and sell it. And it's very difficult to do that. But if you'd like to purchase it, go to the website. I guess I do have a conflict of interest. So I want to talk next or kind of end on some of our more recent work on solid tumor diagnostics. So I mentioned that I wanted to use quantum dots for that earlier. I found out that's not really how they do testing. And I sat on that idea for many, many years, trying to think about how I can help with solid tumor diagnostics, because that's a larger patient population that have a solid tumor. And I eventually came up with something. So this is a collaboration with Jose Otero in neuropathology, Ben Gherkin in bioinformatics, and Carlos Castro, who's in our mechanical engineering department. So solid tumors do not use fluorescence. And the reason is because tissues autofluoresce when they're fixed with formaldehyde, which most of them are. And this is a good example of that. All of the red in the background here is false signal. And if you look in the white circles up there, that's the real signal, and the red circles are red blood cells that are bleeding through. So I can't really tell the difference between my false signal and my real signal. And if I'm diagnosing a patient, I really need to be able to tell the difference. So instead of fluorescence, they use colored dyes called chromophores to label biomarkers. So this is HER2 again being labeled in a breast cancer biopsy sample. And if you're in heart and you mix all the colors together, what happens? You get brown or black or whatever. So the problem here is with fluorescence, I can excite it one wavelength and just get red. I can excite it a different wavelength and just get green. I can deconvolve those signals. I can't really do that very easily with these colored dyes. So what they do is they label one antigen per slide. So you have a tumor sample, you section the sample, slide 1, I label for 2, slide 2, I label PR, slide 3, I label PR. And that's been okay, because initially with our personalized medicine schemes, one biomarker by itself was suitable for triage. But as technology evolves, it starts to become important whether you have A, B, and C and whether they're in the exact same cell. And I can't answer that question with this method because I'm only labeling one biomarker per slide. And even if I label A, B, C, each slide is sufficiently thick, it's a different set of cells. So my problem that I'm trying to address is how can I label multiple antigens in the same slide so that I can get these multiple markers like we were talking about for the AML and leukemia, if you're labeling 8-9 things, how can I do that? And so we were inspired by the idea of erasable labels. So this is one example from fluorescence pathology. So again, not for solid tumor, but what they do is they label a cell and then they can use chemicals or light to either degrade the dye so it kind of chemically falls apart, or they can have it be removed. And so now I can label it again. So I get rid of it, and now I'm free to come in and label with a second set of antigens. So that was the idea. But this uses UV light and harsh chemicals, which can damage the tissue. So that approach isn't going to work. We also looked at this approach, which was based on DNA. So here you have a DNA molecule that has a fluorophore on the end, and you're using it to label a sample, and you can erase it by adding complementary DNA. So it kicks off one of the strands that has the fluorophore, so the signal goes away. And now I can come in and label something different. The problem with this is it only has one dye molecule per DNA strand. And when you label antigens here, you usually have a primary secondary scheme. So you have a primary antibody that might have like 6 secondaries on it, and you're getting this amplification of fluorescence signal, but you don't get that here. And this also, the dye is not environmentally protected. So it's fine in cell culture, but when you start getting into tissue environments, there are things in those environments that can degrade those labels. It doesn't work very well. So our question was, and there's only, so only one DNA per strand, it doesn't have the implication. Could we do something else? And I saw this paper when it came out in 2017, and I was just really inspired. So this has nothing to do with what was going on there. What this group had done is they were using something called a DNA tile. So there are 3 DNA strands that come together to form a tile that has a triangle or Y shape. And then that tile can self-assemble into hexagons. They had taken this and put it inside of a liposome, and it was an artificial cytoskeleton. That's what they were doing. I was like, hey, could we use that DNA to put a cage on the outside of the micelles that I was making for the farm rats to protect dye? So the dye is inside the particle, kind of like what we did with the farm rats. But then I'd have all of these dangling strands that I could use to have more valency to amplify signal. So this is basically the scheme. It's the same triangle that you're studying, but instead of building a cage on the inside of a liposome, we're doing it on the outside of a micelle. The DNA is attracted to the micelle through charge attraction, so DNA is negatively charged because of the phosphate backbone, and the particle is positively charged. So she's going to kind of bind and begin to self-assemble on the surface. And in the interior, we can put either those chromophores that are used for cell labeling in tissue samples, or we can put fluorophores or quantibat. So the scheme is this. I have an antigen that I want to study. I have an antibody that binds it. And so it's going to come in and bind the antigen. And then my particle is going to come and bind that through complementary DNA. But the green is not fully complementary to the yellow. So at this point, I'll have a signal and I can image that. But then I come in with the yellow DNA that's more complementary. It kicks off this particle and I can flow it out. So first we had to show that we were actually building these cages. And so there's 3 different experiments that we did. In this experiment, we had a particle that had dye on the surface and the DNA had a quencher. So when it binds the particle, the signal goes away. So you can see that. Great. The next experiment was the reverse. So we have a blank particle and DNA has a fluorophore, so when it binds, the signal goes up. And we can see that at a certain point it saturates the surface. And then the last experiment is the interlocking experiment, where some of the cages have a quencher and some of them have a fluor. And when they come together, the signal should go away. So you see the signal rise initially because the tiles are not interlocking yet. And then it goes down when it levels off. And that's because of steric hindrance. So not every quencher and fluor are meeting each other. So some of the fluors are not getting quenched. But this basically showed that we were in fact forming these DNA cages. So then we did label erase experiments. So here we are just in solution. We're adding a DNA that has a fluorophore on the end. It's binding cage and we're erasing it. And we did multiple cycles by using increasing base tuning. And we found that our erase step was around 95%, with 5% residual signal. Now we move to cell labeling. So here we have the Integrin beta 1, one of my favorites that we're labeling. And then that is a traditional controlled label, standard labeling scheme. This is with the cages. So it looks similar. So that shows that it works in terms of labeling. And then we did label erase cycles. So here we're now labeling actin inside the cell. And that was because we wanted to show that it could function in an intracellular environment, which is more challenging than an extracellular environment. And you can see that we label it, we erase, label it, and erase. And this was done in a flow cell. So we're flowing in the erased DNA at a certain rate. And then these are blow-ups of kind of our wide fields of the label and erase. So that was all published in our fairly recent paper in Nanoscale, but this data is not published yet. So then you're probably saying, okay, Gabby, that was all fluorescence. She just told me they could use fluorescence. So we started with that because it's easy for our lab to do. And then we increase in complexity. So now we're looking at tissue. And one of the first questions we had to ask is whether these cages would be compatible with the detergents that are used in tissue processing. So Triton-X 100 and Tween. And so we repeated the label erase and Triton and Tween, and we saw that we still got the 5%, so they are compatible. Interestingly, if we take these same micelles without a cage, and put them in Triton and Tween, they dissolve. This is detergent, and it's a possible, that's what it does. So we didn't realize that the DNA would also stabilize the structure, but that was a nice unintended benefit. We then asked whether they could penetrate a tissue specimen. So this is the tissue specimen that's 40 microns thick. This is just the brain slice. On the left, we have intensity. And you can see the middle is has very high signal, and the left and right have a low signal. That's because this is a floating section. So we floated it with the cages for a while, and then we transferred it to pure water. So the edges are, the dye is diffusing out, but the middle, you can see it didn't fully penetrate the slice. We then asked whether it damages tissue. So these are just images of a tissue specimen before and after label and recycle. And we use machine learning to basically compare them, and there were no differences between them. So we don't see any changes in tissue architecture or damage resulting from our methodologies. So then we did the experiment, and that was 3 years right there. Just for all the people who are having slow projects, like sometimes you have to be patient. So this is what the tissue looks like in fluorescence with nothing done. This is the negative control, where we add cages that they're not specific for what we're labeling. In this case, we again labeled actin because it's expressed in every cell, so we'll get a nice gray signal. And then here's our experiment. We were able to label it, and I guess the erase is on the next slide. Then we did do the chromophores. So this is the control. This is what they use now, TAP. And we're looking for a signal on the edge of the slice. So this is the brain slice, and we're labeling striations. And this is the green label. And so you can kind of see it's kind of working. It's not bright enough. When I showed this to my path collaborator, he's like, you need to do brown, switch to brown. We're working on this. But we do have evidence that it's working. And then this is the erased samples. You can see there's just nothing. So we're pretty happy with this technology. and think that it may allow clinicians to do multiple labeling in solid tumors. So we have some preliminary evidence, but definitely more work ahead of us. So I just want to bring it home and kind of remind you, I'm really excited about diagnostic technology. I think this is the way that we get to better cancer outcomes. And I am a breast cancer survivor myself. 13 years, I was diagnosed at stage 3, so I'll let you cheat on that. So this is personal and it means a lot to me. This is my group. So for this project, we have 3 that have left the group, graduated, so that were involved with Silvio. De Arroyo Fernandez is now a research scientist at Nationwide Children's Hospital in Columbus. Mark Calhoun is now employed at Natera, and Elizabeth Jurgens is actually a new faculty starting this year at Ohio State, so we're excited about that. And then we also had contributions from Nandita Gayatri Srivanesan, and yeah, I think that's it. Oh, and Paul Lee helps with some of the tissue work. So, thank you. I appreciate the invitation. I'd be happy to take any questions. Remind you of all the different things.
At this time, open the floor for questions for Dr. Winter.
Yeah, that.
Wait to see if anybody else had questions, but I'll go ahead. It's very interesting, really great work. Thanks for sharing it with us. I had 2 questions. First of all, on the pressure study, it's really interesting results. I don't know if that's been shown before that there's no parabolic response. I'm familiar with the damage to the cell viability at the lower pressure, but I haven't seen it shown with high pressure. What was the time point? What assay did you use to measure the viability?
That's a good question. So we exposed them to pressure for 6 hours.
Okay.
So we did a 1 hour time point, and we actually don't really see any differences in 1 hour. So we did sort of increasing until we started to see differences, and then we went forward with that time point. But that's a really good question because we don't know. And we tried some different experiments, and these take it out long enough, all the cells do die, right? But that's also true of people. You take your infinite time point, that Kaplan-Meier is always 0.
But it's pretty static, the interstitial pressure is pretty static, or does it fluctuate?
So it would fluctuate with blood flow, and that is one thing we haven't built into the model. So this is a pretty crude model, and then obviously in GO models as well, we're just trying to bridge the gap between that. But yeah, and it wouldn't be very difficult to put that on like a peristaltic pump and have to mimic better.
Yeah.
The pressure.
And another question, very minor, but that last slide you showed where it was green and they said need to be brown. Can you just pseudo color it, or do you have to actually redo the
That's a
chemistry?
great question. I guess you could. What we're trying to do, so I learned from starting the company, clinicians don't like change. They want it to work, I don't like change either. They want it to work exactly the way that it works now. So if you can come in and say, my brown label is the same as the brown label you're doing now, only it also does this, they're more likely to adopt it. Whereas if I have to teach them about pseudo color software, like it starts to go downhill.
So they want to be able to take the slide and put it right in the microscope and not have to do software.
Yeah.
Right?
Well,
Because
they
you
don't
can
have
easily
to
change.
use digital pathology, but they're also very sensitive because pathology is really an art form. I mean, when you look at some of these top slides, they'll be like, blah, blah, blah. I'm like, I don't even know. It's like a Rochefort ink blot or something. I don't know how they see. And so they're very sensitive to any computer manipulation. And I'm respectful of that because I want to make tools that they'll actually use. And the reason that we did green, we have a red one as well. So we started with red, and that worked great. And then he said, no, that's blood. Blood is red, can't do red. So we went off and did green, and then brown we've been working on, but we've been having trouble encapsulating enough brown to really get a good signal. So we're trying different brown dyes, and this is one more better than that, and it's an effort, but we'll get there eventually. Yeah.
So I know you had some software AI integration Can you tell me a little bit more about, that process for taking a look at the different slices? It was one of the
Yeah,
middle
so a
experiments.
lot of that work is done on my image analysis collaborator, so I'm a little bit more agnostic. But I know that they use algorithms to, so for just the stacking, what you're doing is you're doing confocal, so you have a stack of images. You can compress them all into one image, you can do that in ImageJ, and then start extracting features. So that's the part that I can probably do. And then our collaborator does supervised models, because unsupervised needs some more error, and he has a paper on that. And I think I'm on one of them, even though, like, I understand, but not at the level, obviously, that he does. And he has technologies that can identify features, so he can find a cell, and then he can find the nucleus, and then he can pinpoint different particles. So then he looks at how many pinpoints there are and will count them. So he basically has technologies that would do that.
Okay, for the feature extraction.
Yeah,
portion.
and it's 5 pixels, so you are kind of limited. But like within each pixel, you can do a point spread function to identify the centroid, which gives you a little bit better resolution. That's about the extent of my knowledge.
Gotcha.
And then machine learning was also done by our neuropathology collaborator, because he's developed algorithms to identify different cell types and look at differences between tissue slices and machine learning. So that's his technology. I don't. Yeah,
Talking about a liquid pathology and a solid pathology, what do you find different between the two? What do you find basically the same thing?
it's completely different. There are different people, different groups, different diseases. So liquid pathology is basically a clinical flow cytometry lab. All pathology will have not only cancer, but other cases coming to them. So our liquid path group also does HIV, malaria, I think disease, the whole, anything that's blood-based is something they might be looking at. And then your solid tumor path has not only cancer, but any other sort of pathology that might be, I had a polyp removed in my colonoscopy, is it benign or not? So the workflows are entirely different. The liquid path is blood, and they might start by removing the red blood cells and then analyzing the remaining cells. Everything is done in liquid. The solid tumor path it has 2 workflows, frozen and FFPE, formaldehyde fixed paraffin, and frozen is typically done during the operation. The question is, did I get everything I was supposed to get? So they'll take a piece of the tissue, they section it off, rapidly freeze it, immediately go look at it, and then they give an answer back to the surgeon. Often they're still in surgery, the patient's still open, and they need an answer right away. And then the FFPE, they fix the tissue so they can process it at their leisure, and it takes about a week. So it's going through all the different steps of sectioning it, imaging it, and looking at it. So they're just completely different workflows, and the types of people and their personalities are different. Yeah. I don't know if I, yeah, okay.
Any other questions for Dr. Winter? All right, let's give her a hand and thanks for coming to talk. Please take a moment, the QR codes on your desks to sign in for today's seminar through your cell phones.